Creutzfeldt-Jakob disease, CJD, is a neurodegenerative disorder belonging to the group of transmissible spongiform encephalopathies. The transition of physiological soluble neuroprotective prion protein, PRPC, into its insoluble misfolded isoform, PRPSC, remains a central pathogenic event. The progressive accumulation of isoform PRPSC within the brain tissue results in spongiform degeneration and a plethora of clinical symptoms. Typical CJD manifests as progressive dementia with myoclonus, visual or cerebral dysfunction, or mutism. However, a growing number of studies indicate that CJD may present with psychiatric manifestations including hedonia, anxiety, irritability, depression, insomnia, psychosis, and catatonic behavior.
PRPSC, the abnormal form of normal prion proteins, are known to cause diseases called transmissible spongiform encephalopathies, or TSEs. Neurodegenerative disorders characterized by long periods of dormancy but rapid progression once onset of symptoms begin. The human TSE diseases include Creutzfeldt-Jakob's disease, Kuru, and fatal familial insomnia. Abnormally formed prion proteins can exist in two forms: an inactive soluble state and an aggregated form that is capable of self-perpetuation. The self-perpetuation of the abnormal prion protein occurs catalytically, analogous to that of an enzyme binding to its substrate. In our case, the substrate is the normal form of the prion protein. In order to determine if a protein was the agent causing transmissible spongiform encephalopathies, researchers performed stereotactic surgery on prion protein knockout mice and injected them with the abnormal prion proteins. In turn, the mice showed complete resistance to the abnormal prions. It is currently theorized that different abnormal prion strains are the result of the presence or absence of cofactors during the conformation of normal prion proteins into abnormal ones. The transmission of Creutzfeldt-Jakob's disease can occur genetically via the inheritance of a gene mutation, iatrogenically through contact with infected medical equipment, ingestion of tainted meat such as beef with mad cow disease, and by direct or indirect contact with infected brain tissue or cerebrospinal fluid. Aerosolized abnormal prions are theoretically thought to be capable of causing infection as well. In 2003, a British patient died after receiving blood transfusion from a donor with Creutzfeldt-Jakob's disease, showing the pathogen is also capable of being transmitted through the blood. Prions are also one of the most difficult pathogens to eradicate, boasting an incredible resistance to heat, radiation, chemicals, and prolonged autoclaving. In the bottom right corner, an image of the prion protein's structural organization is provided. Prion proteins are also highly concentrated in areas of high synaptic density. In mice, the prion protein is found in the highest concentrations in the stratum radiatum, stratum oriens, stratum lacunosum moleculaire, and the molecular layer of the cerebellum. The combination of abnormal prion proteins leads to characteristic lesions in the brain tissue that you see before you. The dense pursal clusters you see are neurons. In the case of our patient, she was tentatively diagnosed with Creutzfeldt-Jakob's disease 9 months after the onset of symptoms. To definitively diagnose a person with Creutzfeldt-Jakob's disease, one would have to perform a brain or nervous tissue biopsy. Autopsies of brains with Creutzfeldt-Jakob's disease reveal enlarged astroglial cells and large aggregations of abnormal prions, which are thought to be toxic to neurons. Due to ethical concerns, most diagnoses of Creutzfeldt-Jakob's disease occurs post-mortem. Part of what makes diagnosis of this disease so difficult is due to the aggregation of abnormal prions not eliciting a host immune response.
First discovered in the 1950s, Kuru is a rare and fatal nervous system disease. Its highest prevalence occurred among the 4 people in the highlands of Papua New Guinea. 4 people contracted the disease by performing cannibalism on corpses during funeral rites. Women would eat the brains and also feed small amounts of brains to children. The study of Kuru has helped along scientific research in a number of ways. It is the first neurodegenerative disease resulting from an infectious agent. This has led to the creation of a new class of disease to include CJD. In our case study in 2016 in Krakow, Poland, we have a 49-year-old woman who was admitted to a psychiatric hospital for the first time 9 months before the proper diagnosis was established. This was due to her bizarre behavior, deterioration of social interactions, reduced food intake, hallucinations, psychomotor retardation with periodic episodes of agitation, passive negativism, and sleep disorders. All symptoms lasting for a period of 4 weeks indicated an acute psychotic disorder, and the patient was discharged. After the discharge, the patient's family observed a deterioration in her health. She ceased to be independent and required assistance with basic activities. There was also a significant deterioration in her vision. Due to lower mood and psychomotor retardation, the patient was admitted to the psychiatric hospital for the second time. Her symptoms remained stable except for increasing muscle rigidity. An electroencephalogram (EEG) test showed pathological tracing with diffused and generalized paroxysmal theta activity. She was discharged with the diagnosis of drug-induced Parkinsonism and treated with biperidone 2 mg 3 times a day. A month later, the patient was again admitted to the psychiatric hospital because of aggressive and agitated behavior. The patient's condition gradually deteriorated further with mutism, increased hypertonia, and negativism. The clinical picture tended towards a suspicion of the catatonic syndrome. Finally, due to unclear clinical presentation, the patient was admitted to the clinical department of adult psychiatry at the University Hospital in Krakow in a state of conscious requiring differentiation between stupor and vegetative state. The physical examination revealed eyes opening spontaneously and no verbal contact, but the patient responded with a grimace to painful stimulation. No spontaneous movement, hypertonia, and muscle atrophy of the upper and lower limbs were present, but because of dysphagia, the patient was supported with a feeding tube. A consecutive neurological examination identified spastic quadriplegia and jaw muscle spasms. The clinical picture of progressive encephalopathy of unknown etiology suggests further diagnostic evaluation towards CJD. Another MRI revealed diffuse linear cortical enhancement at diffusion-weighted imaging sequence. The neuroimaging result prompted the team to consider a CJD diagnosis despite a lack of typical changes in the basal ganglia. The patient's consciousness with no verbal contact was then admitted to the neurology department at the University Hospital in Krakow. There, another EEG showed generalized slow activity without rhythmic slow waves typical for CJD. Because of the lack of typical CJD findings from the EEG, an MRI was suggested, and it showed diffuse encephalopathy, so a brain biopsy was performed after obtaining the approval from a family court. The tissue was treated accordingly to the universally accepted methods for handling tissue suspected of being infected with a pathogenic prion strain, including 1H immersion in formic acid. Histopathological investigations, including immunohistochemistry, were made on sections of samples embedded in paraffin blocks. Staining showed vacuolization, indicating spongiform degeneration. Immunohistochemical staining for PRP demonstrated immunopositive deposits around spongiform changes resistant to autoclaving and proteinase K digestion. These results confirmed the diagnosis of CJD in our patient. At this time, treatment options are scarce to non-existent, and unfortunately, there is no cure for CJD.
Reports indicate that CJD, as well as other prion diseases like fatal familial insomnia, may manifest as catatonia. Our case report and collected data point out several features facilitating differential diagnosis between catatonic behavior observed in mental disorders and that observed in CJD. Those features include short duration of illness, rapid deterioration, and no improvement in psychiatric symptoms despite pharmacological treatment. The presented case indicated that CJD should be considered in the wide differential diagnosis of catatonic behavior, especially when there is short duration of psychiatric symptoms and a resistance to treatment, as well as rapid deterioration of patient's health. Possible cause of CJD was not determined with this research.